Hello and welcome to this webinar, What are the drivers and challenges in therapeutic antibody discovery? My name is Megan Thomas and I'm the multimedia editor for DDW and your host for today's webinar, which is being sponsored by Biorad. In this webinar, you will learn the drivers and challenges in therapeutic antibody discovery, how and where we are seeing innovation and evolution, the impact of antibody engineering, and the potential of antibody-drug conjugates for treating disease. You will hear from Aron Knickerbocker, CEO of Aulos Bioscience, and Richard Yu, CEO and co-founder of Abalone Bio. Over to you, Aron.
Hello, I'm Aron Knickerbocker, President and CEO of Aulos Bioscience, and thanks for inviting me to take part in today's webinar. So we are in a very exciting environment for innovation and continued evolution as it relates to drug discovery. And from my perspective, one of the most exciting areas is the applications of artificial intelligence to drug design and drug discovery. This talks about how we see innovation and evolution. And at Aulos, we're focused on monoclonal antibodies. And monoclonal antibodies are very tried and true approaches to drugging different targets, and they're known to be developable because they're essentially what our body makes on its own when we need to combat, say, a viral infection or something like that. We make antibodies. So therapeutic antibodies are well established for a number of disease states, including cancer, and we know how to make them at large scale, and they're well behaved as drugs once they're used in patients. But what we're doing is employing AI in drug discovery and creating antibodies with the help of AI, married with human ingenuity, of course. So what we do, or what's been done by our collaborators at Biologic Design, who created the antibody that we're now developing, is to use machine learning algorithms that have been trained on data sets of millions of antibodies. So these are real antibodies that bind to real targets, and the machine learns which amino acids and which positions on the antibodies are likely to confer high binding selectivity and specificity to a given target protein sequence. So it's a way to sort of augment what is already created in nature. These are human antibodies, but the machine learning algorithms or the AI platform is suggesting certain changes that can be made to further improve the binding to the target. And that training occurs because there's pattern recognition that the machine learning algorithms have picked up. Much like ChatGPT or some other AI engine does, the patterns get recognized both on the antibody side and on the target side for what is likely to confer a good binding antibody. So in the end, what you create is a human antibody that might have just a few changes suggested by the AI platform. So an antibody is almost 900 amino acids, and the AI might suggest, say, 3 changes to that sequence. But those 3 changes can confer some incredible properties. All that said, true innovation and the evolution of drug development has to occur with people. And we have a great team at Aulos, and I think in any case, what has to be done is to design the right applications of the tools, in this case, the AI platform. So what is the problem that's trying to be solved? And how can that AI platform or that tool be best applied to solve that problem? And it's not like the AI does everything. That's not the case at all. It's simply making suggestions, and then it's up to humans to take those antibodies into the lab based on those suggestions and to test them for which antibodies have the best properties that really meet the target product profile that's sought, which is to design an antibody that binds to a very specific target. And in our case, it's a very small target on a protein. So we need humans to figure out what's good, to keep that, and discard the bad. And coupled with the AI platform, it's a powerful way to design human antibodies. What is the impact of this antibody engineering that we can do with the AI platform? Well, as I mentioned, we can optimize the binding regions of these human antibodies. And this really mimics what our immune systems do naturally. When we need to make antibodies, say, to battle flu virus or something like that, our immune system does a process called somatic hypermutation, in which lots of changes get rapidly introduced into template antibodies that might kind of bind to the target to try to improve upon that binding. And that's what's done with the AI and ML platform that was used to create the antibody we now have in the clinic. So we're really mimicking in silico what our bodies do in vivo, which is to try lots of changes and see what works best, and then to select that antibody for expansion and production as a monoclonal antibody. So a very specific antibody that recognizes a specific sequence on the target protein. So in so doing, it may allow us to actually redirect human proteins, and in this case, proteins that bind to immune effector cells or cytokines. So cytokines are cell signaling molecules. It's a Latin-derived word. Cyto means cell, kine means movement. And what we're trying to do is to get cells to move into an area, in this case a tumor, and to be active against those tumor cells. So really trying to drive immune activation. At the same time, though, keeping the cytokine away from the immune suppressive cells and the blood vessels, which can result in toxicity. So we really want to drive the expansion of effector cells that are capable of attacking the tumors and killing them. So by binding to a specific part of a very tiny protein, a cytokine, we're able to redirect where that cytokine then binds, allowing it to expand not only the effector T cells, but also the immune memory cells, so that the immune system learns, when I see that protein that's expressed on that tumor cell, I'm going to kill that tumor cell. And that's what leads potentially to long-term durable responses, even potentially complete responses, meaning the tumor is eradicated by the immune system. And something that people don't often realize is we get cancers all the time. They're small neoplasms, and the immune system sees them as cells that are somehow aberrant and clears them. It's only when enough mutational load builds up and those cancer cells can escape the immune surveillance that things go awry. So we're trying to tip the balance back to an immune activated state whereby the effector cells recognize those tumor cells, kill them, and the memory cells remember that if they ever see them again, they will kill them again. And so that's how you can get these durable long-term responses. So this has really allowed us and other scientists, I think, to rethink how we approach certain drugs or certain targets that were previously written off. Because cytokines have a variety of effects, but if you can really tune it by redirecting, it may allow for the production of an immune response against the tumor. And others had tried, by the way, to create antibodies to the same cytokine to which we've now developed this product, and failed using traditional antibody techniques. It wasn't until the AI platform was applied that success was achieved, and we have now an antibody that's highly specific and allows for the redirection of a particular cytokine. One of the things that's very important, I think, is really to follow the science, and this has guided me throughout my career. I think that's been why I've been associated with positive outcomes, not just by me, but by my teams and by the companies at which I've worked. We follow the science. And I think that's still key, regardless of the platform or the tool that's being employed, whether it's AI or not. You still have to have something at the drug level that is a novel and compelling mechanism of action that can drive some new effect, some new clinical benefit for the patients. And then when you take that into the clinic, it's very important to run a well-designed trial, to ask the right questions, to optimize for dose and schedule, to test in the patient population that is most likely to benefit, and to be able to monitor how that drug is performing in the clinic, in the patient. So at the biophysical level, is it binding to the right targets? Is it having the right effects from a pharmacodynamic perspective? Does it have the right lifespan in the body from a pharmacokinetic perspective? Is it toxic? Is it safe? And above all, is it helpful? Is it efficacious? And can it reduce the tumor load or eliminate the tumor in that patient? So really following the data is critical. It's not just about the AI platform. It's about doing well-designed clinical trials and generating quality data. And that's what eventually and almost certainly will advance a successful product if the data are of high quality and there's clear clinical benefit with acceptable adverse events. And one of the things that's been seen time and time again in immunotherapy for cancer is that if there's activity in one tumor type, there's often activity in other tumor types, that these are generalizable outcomes across a range of different cancer settings. And that's what we hope to achieve as well. Just to come back to some of the key takeaways today. With respect to innovation in antibody discovery, for years, the same sort of approaches have been used to generate antibodies. And those were very effective in many cases, but not in all cases. And in the situation that we've tackled, we've drugged a very tiny protein and a very tiny portion of that protein. And it wasn't until the AI platform was applied to this question or this problem that was quite vexing that we were able to solve, and our partners at Biologic Design were able to solve and create an antibody that was very specific to a tiny cytokine. And in so binding would redirect that cytokine to achieve the outcomes that we want immunologically. So again, expanding the effector cells that are capable of attacking and eliminating tumor cells, not expanding or activating the suppressor cells, not causing the vascular leak that's known to occur with certain cytokines. And that's been now achieved because we've made this very highly selective antibody with the functional properties that were sought. And this could not have been achieved, I don't think, certainly not as quickly without the application of the AI platform. Do data-driven development. It may seem obvious, but it's very important to design clinical trials appropriately so that the right questions are getting posed and the answers can be realized, whether positive or negative, and then that data should be followed. So we've had surprises in our clinical trials, but sometimes you follow that surprise and it points you in the right direction. Less may be more, for instance. That's one of the things that we've discovered in the course of our clinical work. So really being data-driven, dispassionate about that data, and following it to make sure you're delivering the most patient benefit and hopefully transforming care. And then broad applications. Using AI to do antibody engineering may allow us or others to create antibodies that have the desired properties and the desired effects on the immune system. And what we know from immunotherapy is that if a product works in one tumor type, it may well work in several others, that there's a generalizable phenomenon of immune surveillance and immune control that can be achieved with an active immunotherapy. So that's ultimately the goal as well, is not just to help patients with one type of cancer, but hopefully many patients with many types of cancers. Thank you very much.
Thank you, Aron. That was really enlightening. Next up, we'll hear from Richard Yu, CEO and co-founder of Abalone Bio.
All right. Thank you, Drug Discovery World, for this opportunity to speak with the audience today about drivers and challenges in antibody discovery. I'm going to focus a bit today specifically on a particular class of antibodies, functional antibodies, and how we're trying to bridge that gap. My name is Richard Yu. I'm the CEO and co-founder of Abalone Bio, and we're a company devoted to finding antibody-based modulators of challenging targets like G protein-coupled receptors. I want to start off with a quick story. Saw this on YouTube recently on the channel that I follow. There's brought up this idea that ChatGPT is not able to draw a full glass of wine. I thought this actually had some lessons to teach us about functional antibodies and how to discover and design them. So it actually has a difficult time trying to draw this. I tried this out myself, and here are some examples. You know, if you try to generate a full glass of wine, you get that. Okay, try to give it a clue by saying full to the brim of the glass. Skills gives you something like a half glass. It knows what full glasses look like because you can generate a picture of a full cup of coffee and even an overflowing one. And so, okay, well, maybe it's about the definition of full, and we want to just be very clear about how it can be overflowing. Of course, that's what it shows with the overflowing cup of wine, still that half cup of wine at the base of it. And then even if I try to bridge that gap by putting those two concepts in the same exact prompt, it still has a partially empty cup of wine. Like, super interesting. And just quickly looking at Google Images and the data out there, it's very clear what one of the major sources of the problem is, that it's just the wrong kind of data for that type of product, right? So if you Google for full glass of wine, you get a whole bunch of images that look like that. Okay, so what does this teach us about antibody drug discovery? Well, the amount and the type and quality of the data that you're measuring is important. It's especially important for antibody drugs, and it's especially important for functionally active antibody drugs. And when I say functionally active, I mean antibody functions that go beyond just binding and potentially sterically occluding a ligand and getting in the way. So that's sort of easy or trivial case of inhibition, but actually pharmacologically active. Those things that modify the structures and ultimately the functions of the targets that they bind, so that you get a change in, say, a signaling property that you're looking for that can help treat disease. Okay, so there's a huge driver for this. The functionally active antibodies, if you can find them, address major challenges in drug discovery. So for many of these targets, such as the G protein coupled receptors that we're interested in, specificity is a huge issue, right? You can find drugs that bind them and activate them or change the activity in a way that you want it, but they have tons of off-target effects. They light up a whole bunch of other GPCRs and potentially even non-GPCR targets. Antibodies, of course, are very large and structurally and chemically complex. They have the potential to be exquisitely specific. Antibodies also address the problem of bad drug-like properties from some of the small molecules and peptides, whether it's stability, half-life, solubility, poor metabolism. Antibodies are quite stable. They have long half-lives. They're very soluble and are processed quite healthfully, reasonably by the human metabolism. And of course, activity for certain targets. The small molecules just can't quite engage all the necessary parts of the target that you need to facilitate the effect that you want. And antibodies are very large and complex, like I said earlier, so they can engage these multiple parts and fully activate or otherwise modulate the activity of some of these targets. Now, there have been some great successes in this field. For example, for receptors that need to dimerize or heterodimerize to activate, like the receptor tyrosine kinases, there have been some great examples of activating or modulating antibodies. Of course, for some of the other targets, like G protein coupled receptors, ion channels, these types of molecules that are pharmacologically active are actually very hard to discover still and create. So here's an example of a GPCR, and you want the antibody to come and sort of bind the top loops, the extracellular loops, and arrange a structure of the protein to give you a function that you want. Well, this actually is not a simple lock and key type of problem. If you actually look at these proteins, there's a molecular dynamic simulation of the GPCR. You have these 40, 50,000 atoms jiggling around with tons of conformations being sampled. So it's actually quite difficult to determine what antibody you need and what structure you need to bind to give you the functional property that you want, right? So antibodies that can bind in just the right places to give you the structural changes that you need on the inside of the cell, in the case of a membrane protein receptor target, are rare, right? There are just so many more ways that you can bind this in a trivial way that doesn't do anything. And it's also not just a matter of mimicking the agonist or the natural ligand, right? You can imagine there are all these extracellular loops and bits and parts that you can bind to functionally, internally cause the same effect as the binding of the target in an orthosteric way. But binding allosterically, you can get a similar effect downstream. So it's a very complex and hard to solve problem that we're asking these antibodies to solve. So this ends up being primarily a huge search problem. It's a difficult search problem because these antibodies all are very similar, right? So you're sifting through a huge pile of keys in your junk drawer to find that one that opens up the lock that you need to open up. Okay, so typical discovery is really focused quite a bit on reducing those numbers. And the primary way of doing that is affinity, right? So no matter how you start, you generate your initial antibody diversity, whether it's through immunization of an animal or just an in vitro library, you start off with large numbers, hundreds of millions, billions, even more than that, and you narrow down to a number that's physically capable, that it's easy for you to handle. And that typically involves something like affinity enrichment via phage display or yeast surface display. Then you end up with a high affinity subset. Makes sense. You want your antibody to bind for it to be functional. and then you run activity assays on those to try to find molecules that have the activity that you want. But the problem is affinity is not the best way to yield activity. Of course, you need to bind, but binding tightly is not as important as binding in the right place, right? So if you focus maniacally on high affinity molecules, you end up getting things that bind really tightly and don't do what you want to the function of the target, right? It's like grabbing scissors like that, typically binding some immunodominant epitope. What you really want to do is bind all the parts of the protein you want, whether it's a mimicking orthosteric binding of a peptide or allosterically grabbing onto those loops and just sort of massaging the protein structure in just a way that allows intracellular signaling to happen. That's what you really are searching for. So you really want to do functional assays in your search. AI will not save us on this, right? So AI has made some great strides, obviously in structural prediction with AlphaFold and all the derivatives and even some antibody-specific folding structure prediction programs. And there's been a lot of really cool progress lately in antibody binding. So even de novo, in some cases, being able to predict sequences of antibodies that can bind a target of interest. But that next step, going bridging to function, has not really been bridged yet. We have one example of a prediction of an antibody that's active against a GPCR, and I believe that's the first one that I'm aware of, having a functional agonist against a difficult target like a GPCR. Large-scale functional data is the key to getting this next stage, to getting to function. And it's necessary both for discovery and for design, right? One really cool thing, again, we have some data showing an example of this, is that a well-trained AI model can really expand the searchable sequence space in silico. So our platform can functionally test about 100 million antibodies in a given experiment. But when we trained an ML model with that, we were able to, in effect, search a huge sequence space, even for a local optimization around a particular hit, right? So in that case, we're dramatically expanding the capacity beyond anything that you could hope to achieve with experimental wet lab-based techniques. So, okay, so we really want this data. How are people doing this today? Well, there's, of course, is the brute force conventional methods. You take robots and multi-well plates and armies of people, and you just start crunching through these assays in low throughputs. There's been quite a bit of advances on the technological side in terms of miniaturizing these assays. So using either microfluidic channels or droplets where you can encapsulate one or two cells, they've been able to, for example, isolate B cells that produce antibodies from say, an immunized animal, and then in the same chamber or droplet, trap a reporter cell so that you could test the activity of that secreted antibody. Super cool stuff. And with that, you can test orders of magnitude more antibodies in a given sort of campaign. And then more lately, such as what we're doing at Abalone, we've engineered cells to individually make and test antibodies against a particular target of interest. And in this case, you sort of take advantage of in our case, using microbial cells, yeast cells, take advantage of the high numbers that you can cram into a test tube or into plates. So you can get some really, really high throughputs and start getting at library scale functional assays. Okay, some quick things to think about in terms of the focus of your data. So of course you have something like AlphaFolds or like essentially general protein folding prediction algorithm. And then you have things that are much more focused, right? And for example, we focused on GPCRs, and we think this is a nice sort of balance because we can measure a lot of data, but it's in a space where there are many structurally conserved and similar targets. Right? So we can translate more easily, hopefully translate data and learnings from one receptor to perhaps a new target without saying something we're going to solve functional antibodies against all possible membrane and cytosolic proteins. Then your possible space becomes so large that your data that you're measuring is so sparse that the machine learning can't really fill in the gaps there and build a model around that, a predictive model around that. Also thinking about strategy, how you want to use this, the tools that you develop. Where are you on the experimental to de novo / computational spectrum? Obviously, I think the place where you want to be is something where you're using a bit of both. It's quite a bit of talk about how sexy it is to be able to do click a button on the keyboard, complete de novo prediction. But actually, if you have experimental data, you might as well use it. I mean, ultimately, we're here to find drugs, not make something in a computer without experimental data as a goal in and of itself. Okay. Zoom through a lot of this, but hopefully touched on a few interesting points. In conclusion, for finding these functionally active antibodies, structure and binding information and what's available out there, it simply isn't enough. We do need more functional data. AI will not save us yet. That also needs more functional data as well. But once we do that and we integrate that with other types of data, not only in terms of the different kinds of antibody data, such as developability or stability or immunogenicity, I think integrating across the sort of workflow is also going to be very powerful, right? So for example, you could use this for discovering antibodies, but then a trained model can also help generate a better starting library, right? So you can start getting some very virtuous feedback loops that can really accelerate this process. Okay, I hope that was at least touch upon some issues here. Please, if you have any questions or want to discuss any of these points further, I would love to talk with you. My contact info is below, or you can reach out to Drug Discovery World folks for my contact info. Thanks so much for your attention.
Thank you, Richard, and thank you, Aron, for those presentations. Before we head over to the Q&A, I'd just like to remind everyone that this webinar will be available to access on demand on the DDW website, www.ddw-online.com. Okay, we will now move on to the questions. Aron, this first one's for you. How does AI power drug design specifically, and does it speed up timelines for bringing a drug through the clinic?
I think the answer is beginning to emerge that yes, it can significantly reduce timelines for creating an antibody and bringing a drug to the clinic. In our case at Aulos, we've applied AI to a difficult problem of a target that was hard to drug using traditional antibody technologies, whether that was hybridoma technology or phage display technology or animal model technology that creates antibodies. Those approaches have worked very well in the past for a number of targets. They didn't work well for this target. So if you have an AI platform that's been trained on millions of antibodies and billions of amino acids within those antibodies and the target epitope on the binding protein or the target to which it binds, that allows for rapid recognition by the AI platform of patterns and sequences that are likely to confer the sort of target product profile for binding specificity and selectivity that's desired. So I think it's really allowed us to leapfrog quickly, and our partners at Biologic Design have this platform that they use to apply AI to refine human antibodies. Again, it's a human antibody. It's not an artificial antibody. It's a human framework, but with slight tweaks that have been suggested by the pattern recognition that the AI platform allows. So I think it's really allowed us to go faster, to create a product that is a clinical candidate, and now we're well into the clinical trials with that candidate in phase 2. And that's because the sequence that was desired, or the profile that was desired, was just achieved so much more quickly by leveraging AI, coupling that with human ingenuity, human recognition of what is the best candidate to pursue among those suggested changes from the AI. And I think we could not have gotten here at this pace using other technologies.
Thanks for that answer. Another question for Aron here. How is the pharma/biotech industry working with the FDA to speed up the incorporation of AI into drug development and compete with other countries?
Yeah, so FDA has issued some guidance recently this year about using AI in drug development, and what they're trying to do is apply a risk-based framework for AI and its applications to drug development. So they're not saying you can't use AI models. They just want to have a framework to make sure those models are in fact reliable and credible and can be relied upon for regulatory decisions. So whether that's a question of drug safety or efficacy or the quality of the drug, if the AI is being used for any of those things, it's got to be a credible model. I think that's what FDA is saying. But FDA is trying to work with industry. Industry is trying to work with FDA. I'm not an expert on the entirety of this field, but that's my understanding. And I think we'll be seeing this evolve rapidly as AI is now being applied in lots of different ways. As I mentioned, safety and efficacy and quality, but also in discovery, as we've just talked about here with regard to antibody creation. So I think this will be an evolving area of interplay between regulatory bodies such as FDA and innovators like Aulos and others as to how the AI is validated to be credible with respect to these regulatory decisions.
A great answer. Thanks again. And now a final question for you, Aron. What disease spaces may benefit most from advances in therapeutic antibody research?
Yeah, I think we're on the precipice of some very exciting advances in the fight against cancer and hard to treat tumors. And obviously, if we could reduce mortality in even one indication, that's a win. But as noted, immunotherapy can sometimes be expanded or applied to several indications if you can somehow activate the immune system or take off the brakes on the immune system to recognize those tumor cells as really not self and cells that need to be eliminated. So we know this can be achieved in certain contexts effectively. There's obviously been great success with the checkpoint inhibitor class, but I think that's the beginning, not the end of immunotherapy. And AI, I think, holds the promise of allowing us to continue to try to tip that balance toward an immune state or an activated state that recognizes tumors, remembers what those tumor cells look like, and keeps them eradicated. And the research that we're doing at Aulos and our great team is running the clinical trial. This is showing that this is possible, that we can achieve immune activation and really, I think, generate some exciting results for patients as their immune systems begin to take over and control a tumor that could be life-threatening. So I'm very excited about the implications for other studies and future advancement in immunotherapy.
Thanks for that answer. Here I have a question for Richard. What do you see as the biggest challenge in collecting large-scale functional antibody data, and how can we address it?
Yeah, that's a great question. I think I can speak personally. The problems that keeps me up at night is really, are we measuring data that's accurately representing the biology? I can give you a recent example that illustrates this idea of, is your receptor in the right cell type, and does it have the right components to accurately represent the biology in the ultimately the human disease model that or disease context that you're interested in? Something as trivial as having different ratios of components in our example with our G protein coupled receptor targets, different downstream G protein subunits, right, can dramatically change in some cases the signaling properties, right? So, is the data that you're collecting, is that being generated from a relevant model? I mean, that sounds trivial, but I think a lot of people will overlook or can overlook the sort of subtle quantitative differences between the different types of assays for the same exact protein target, right? So I think that's something to keep in mind. When I think about this, it's honestly amazing that any drugs work, just because of how complex biology is, not only at the protein level, but within the pathway level, how they interact with different parts, and in the context of the overall organism. It's like it blows my mind that these things ultimately work.
Thank you. And another question here for Richard. What is one innovation in this space that excites you most?
Yeah, great question. So for me, I'm still blown away by the transformer models and the large language models. I did some protein structure prediction many moons ago. Don't have to say the date, But let's just say it was on secondary structure prediction, sort of in the early days of neural networks. And the idea now that you have these large language models that are sort of pre-trained on proteins or antibodies or some combination thereof, that sort of like understand proteins or antibodies in general, and that you can fine tune that with completely orthogonal data types. Like in our case, we're doing sequence function data. You could have sequence stability, sequence immunogenicity, and sort of combine these all at the same time is just awesome. I think that that is truly a sort of quantum leap in the technology beyond just the important but more sort of evolutionary types of just measuring, having much more data out there for at least for the English or the word-based large language models like ChatGPT or computational power, right? So I think the protein space and the antibody space is catching up. And I think as we pile more of these different types of functional data sets, as well as fill in other gaps in other properties that are interesting for drug development, or maybe even wrapping in later stage clinical data or preclinical data, things that are very sparse downstream, but hopefully if you could incorporate that information early on, would allow you to make better decisions that could potentially save you time by picking better molecules for downstream criteria earlier on. I think that stuff that's super exciting to me. And this is not just about pure AI. I think this is about collecting also the right data and coming up with the technologies to also collect and process and properly treat that data so that AI can take the best advantage of that.
A great answer, thank you. That brings us to the end of this webinar. I'd like to thank everyone for tuning in, and a big thanks to our presenters. If you'd like to listen to the webinar again, it will be available on demand at www.ddw-online.com. Thank you to the sponsor of today's webinar, Biorad. You can now view Drug Discovery World content online on your mobile or tablet. Thanks for listening.